Ron will be introducing his subject, which is, has to do with what I mentioned earlier, which is scaling of SynBio. He has 3 stellar speakers with him on the panel, 2 of whom are online, thanks to our really high-tech setup with hybrid event here. So anyway, Ron, please take it away.
Thank you, Howard. On this panel, the intent is to share with you 2 hypotheses and then see if we can't tear them apart. We want to pressure test them. So one is a little more strategic and the other is a little more practical and or more tactical. And so maybe we could just start with the slides and we'll jump into it. Starting back in, who's handling the slides? Am I?
You are.
Oh, I am.
Come on, 9 years, Ron.
9 years. Look at that. You'd think I could be taught.
He's used to overhead.
Here we go. Starting last fall, I was looking at some technologies, impressive new technologies, the steady stream we've all seen for the past 20 years. And I got to wondering, how have those been affecting the R&D process? And that's what we want to talk about first. Over the past 20 years, the discovery side of finding a new metabolite, a new nutritional product, a new ingredient, a new herbicide, a new drug, a new material versus testing it for safety and suitability for the market and doing the formulation and the scale up and the manufacturing, what we have glibly always said was R&D in the world. Well, if we look at the R, there are a whole range. We could spend an hour talking about massive advances on the R side. Everything from ultra-fast DNA sequencing to robotics, gene editing, combinatorial chemistry. What today we can do in a month would have taken 10 scientists 2 years, 20 years ago. And I started looking at specific examples and trying to put numbers to them, rough numbers. But for example, DNA sequencing, we've all read about that. Everyone knows that the cost has come down by 10 to the 6th since 2001 to sequence a megabase of DNA. So, $10,000 to 10 cents. That's a millionfold. If you look at screening small molecules, when I started in this industry, we would screen 50 new compounds a week. And there were 20 of us to work on it. Okay. Now you can screen hundreds of thousands of compounds in a test tube in a day. And if you go back and look over the past 20 years, guess what? I come up with numbers like roughly 10 to the 4th, 10 to the 5th improvement in screening new molecules to bind to a ligand, for example, or to a cell receptor. Back in October, the Fordyce group at Stanford, and their article actually is what got me thinking about this. They published a microfluidic chip on which you could spot 1,000 DNA sequences with a different amino acid change in each one of an enzyme. And in 4 hours, you could run it and get complete enzymatic kinetics characterization of 1,000 mutants of a given enzyme. 20 years ago, that took a postdoc about a year. And you map that out and it comes out to, guess what? 10 to the 5th, 10 to the 6th, 10 to the 7th, depending upon your assumptions. It goes on and on. 20 years ago, if I wanted to know this 3D structure of a protein, I'd have to get a PhD student to spend 6 months crystallizing it and then do X-ray crystallography, and then I'd have to get mathematicians and statisticians to analyze the data, and we would finally come up with a 3D structure. DeepMind announced 350,000 structures last fall, and they did it in a computer in a couple of months. Again, you're talking many, many orders of magnitude. So then I got to thinking, well, okay, what about on the development side? I mean, this is discovery feeding the product development process, right? So I went looking for examples there. And I had a hell of a time finding them. Some of the best ones I could find, oops, how do we advance this now? There we go. My colleagues at Corteva proudly told me when I asked them that question that it used to take us 12 years to launch a hot new corn hybrid. And now we've got it down to 7 years. That's a whopping 40% increase. Ag microbials, many of them can hit the market in 5 years instead of the 10 or 12 years it takes for a synthetic chemical to get to market because the regulatory processes are a little easier. Synthetic pesticides have really not changed. If anything, it takes longer to get one through the EPA these days. Animal drugs, I've talked to people at Elanco and they tell me that it hasn't sped up. Food ingredients, grass affirmation is a little faster. But basically, the best I've been able to find so far is about a 2x increase in speed on the development side in different areas. So, 10 to the 6th versus 2. That is, to put that in perspective, when I was in Alaska flying a Piper Super Cub on floats at 100 miles an hour, that was the ratio of the speed of light to me puttering along up the Kenai Peninsula. So I sat and looked at that for a while and got to thinking, what does that mean? If this is true and where it is true, what does it mean? Well, it may mean that we've got a hell of a bottleneck coming up in the development cycle. And that leads us to the second topic we're going to talk about on this panel, which is fermentation capacity the bottleneck that we're going to face for most SynBio? So those are the two questions. One is, for this panel, one is, have discovery advances outstripped the human race's capacity to develop products, to turn those into useful products? And secondly, if that has created a bottleneck, is fermentation looming as a big piece of that? Those are the two questions we want to, on this panel, critique, end up with, Ron, you're hallucinating, or yeah, it applies, but here, but not there, in this area, but not that area, et cetera. We want to see what insights we can get out of it. But the intent here is to pressure test these two concepts. I'll quickly say I am seeing, and we've actually invested in some innovations on the development side. Culture Bio, for example, is speeding up the scale up for fermentation, and Debut Bio is sidestepping fermentation and speeding up the development process by going to immobilized enzymes. And there's some other companies doing that. And on fermentation production, there are companies that, like Nobel Foods, and one of the ones I'm involved with that'll announce this shortly, that are beginning to offer production services for proteins in crops. But there aren't a lot of them. Okay, so here's the hypothesis we want to pressure test. And I'm asking, please, everyone else, think about what's wrong with this. Where is it limited? Is it true? Is it not? To what degree? The hypothesis is pretty simple. Discoveries are outstripping development capacity, and therefore innovations on the development side that alleviate that bottleneck are becoming more valuable than new discoveries. And therefore, if you really think about it, and I don't throw tomatoes at me because I spent my whole career investing in discovery, but discoveries we may be going into or already into a phase of human development where innovation to discover new products is so plentiful, fast, and cheap that they're becoming cheap. And the value that we ought to be investing in is on the development innovations, which means guys like me have been investing in the wrong place for the last 15 years. Then we're also going to talk a little further about not only how real and widespread is this, but on the development side, how big is the capacity, the fermentation capacity as a bottleneck to slowing things up? So with those to start, and hopefully having set the groundwork for some interesting debates, I'm going to turn to my colleague here, John Hamer, who is one of the most experienced venture capitalists I know, and I'm going to ask him, on this question of discoveries versus development, John.
Hey, Ron, maybe you should introduce the other panelists who are.
Oh, I
offline.
should do that. I'm
Yeah.
sorry. Please.
Can we put them on the screen, or the other panelists?
Well, okay.
Having a technical issue, so yeah.
That's all right. I'll start. Chris, on the fermentation question, we're lucky enough to have Chris Gusky, who is one of the leading fermentation specialists in the country, with Tate & Lyle for many, many years, with Archer Daniels Midland, retired a couple of years ago, has been consulting and issued with his partner, Mark Warner, recently, a report laying out the case for the coming capacity crunch in fermentation. They called it an apocalypse, a coming apocalypse. We also have Greg Yep from International Flavors and Fragrances, IFF. And I'm hoping Greg will take the opposite point of view, because they have fermentation capacity. They're making it available. And we'll see if Greg wants to tell us that, yes, there's enough of us out here to meet the needs. So that's our panel. John?
Yeah, a couple of comments, Ron, on what you said. Obviously, discovery is the cheapest part of product development. So there's nothing cheaper than sequencing DNA or screening 1 billion molecules in a test tube with a DNA encoded library. The next steps in those procedures, which gene should I use to fix this disease, or which molecule should I use to fix this disease? You're in an optimization and development program, and some aspects of that have been sped up, but most haven't. So development is a very, very different beast than discovery. And so I'd make that point. A lot of development technology also ends up inside big companies, because really the academic community that's doing all the discovery piece has no need for that kind of technology. One example from my background was when I showed up at Monsanto and saw the gene chipper. They have a machine at Monsanto where you drop a corn cob in the top. It strips out all the kernels. The kernels all fall onto these little knobs, little vacuum sort of, what would you call them? Like a little vacuum nozzle. It orients, a blade comes down, slices off a thin piece of the seed. That drops into a microtiter well plate. That gets genotyped in about a day. And suddenly you've genotyped every kernel on the cob, and that kernel can still be planted and grown. Okay, so when you see the machine at Monsanto, it blows you away.
Can you hear me, Kurt?
Because they're genotyping seed.
I can't hear the panel,
before they
so I'm not sure.
And then you realize you're looking at the Gen 1 machine, and they're on the Gen 5 machine, which can genotype 5 different kinds of crops simultaneously. So you can do soybean, corn, cotton. And if you don't think that's hard, take a look at the size of the cotton.
Yeah, where they put that one of the team members is in the asylum closet and has come back and guess what the other people said. So
cycle and be that much faster ahead. Right. And so there's a device.
Actually, I'd rather have audio than video now.
No one
I can at least hear what they're saying.
Developing
I
and
agree.
just seeing that running at Monsanto.
I almost wish I hadn't
blew me away.
closed out the other.
And I'm sure there's
connection I
lots of
had.
developments like that.
Yeah, I'm trying to pull it back up. I couldn't get it. So
I then make a second comment about. So one comment is a lot of innovation around development is tied up inside big companies, and we don't see it. A second piece of it is that we're beginning to realize that bottleneck. And the one place I'm definitely seeing it is in cell and gene therapy. So over the last couple of years, we've had the first gene therapies approved. We've had cell therapies approved in cancer and some other areas. But manufacturing those gene and cell therapies, you can't go to Novartis and say, could you please do this for me? Or to a typical CDMO and say, can you do this for me? You actually had to develop it yourself. And so VC is starting to realize this is a real bottleneck if you want to actually bring drugs to market, which is where you get paid. And so we better start investing in this stuff. And so recently there's been some innovative manufacturing models that have been developed in the pharma industry. Two
I'm not sure what
companies.
happened on the other
One is
one.
called
He kept
Reliance
telling me
and
try to
one's
turn my
called
camera on, and it said,
And what
"Host
those
won't let
companies
you
are
do that."
venture-backed
So
companies that have rolled
Yeah,
up
me
into
too.
manufacturing
Yeah.
technology.
I could hear
They
everything.
then offer the latest in cell and gene therapy manufacturing to a bunch of other companies. And as part of that offering, they get paid and they take a stake in the company that they're doing the manufacturing for. So great investment. You don't have massive clinical risk.
I can
You
hear what-
diversify your clinical risk across your customers, and you've got a revenue generation. So we're seeing that in the biopharma space, particularly when there's really new innovations that have to come to market, that VCs are beginning to realize and invest in that. And I think we see that, and the last comment I'll make, we definitely
now-
see that in food and ag. I was just standing with Arama. I mean, if we're not investing in, if the investors in Plenty are not investing in development, what is it then, right? They're not investing in the seed. They're not investing in, packaging. They're investing in the development stage, really. And the great thing about food and ag that I still stay very excited about is there's a group of investors that you can go to when your company's really early. People like Ron and myself, right? Like we're early stage investors. We're not afraid to take risks. We're going to have a portfolio of risks that we're going to manage. But if that company really starts to work, there's a whole group of investors that are going to come in later and provide capital that can go take you all the way to commercialization, all the way through the manufacturing process, even if that's a brand new manufacturing process. So we have a portfolio company, MycoWorks, that you can go take a look at. No, you cannot have a Hermes purse, just because you know me. Okay, they've got to deal with Hermes and they make purses for Hermes out of their material. So they make a drop-in replacement for leather. Well, we're scaling that company now. So we're building out a huge manufacturing operation in South Carolina, and we've been able to find the investors that are excited about the low carbon, getting rid of the animal product, all the rest of it, that are willing to put money in to sort of build and scale this manufacturing facility. So I think that's one of the really, really exciting things about the food and ag space and why you should be excited to be a part of this community.
I have a suggestion. Louis here from IFF is quite knowledgeable about this area as well. And I'm sure he could say a lot about the same things that Greg could say. Is that true, Louis? Sure. Maybe we could have you stand in for a second.
Yeah, go ahead, Luis.
Okay, thank you. All right, I'm going to try to stand in for Greg and touch on this topic. I think where Greg was going is over the last several years, we've been approached by many companies, and we also approach companies. We have a program to engage with startups. We call it Open Gene to Market. Startups that are doing amazing new innovations, but they want to go to commercial, and we can definitely help there with scale up technology. They often can also use our regulatory expertise to get a new product to the market, something like cultured meats, navigate that ecosystem. And sometimes they may want to tap into our commercial channels as well. Like for instance, food, we sell to all of the CPG companies. We're the largest food ingredient company out there, so we can help on that front, but it's not required. Maybe we just sell it back to them. The issue is spare capacity, spare fermentation capacity. It's actually very hard to find. It happens sometimes. I think in Chris's article, he mentions the Tate & Lyle case where there was a little bit of spare capacity. The DuPont Tate & Lyle facility and Genomatica came in and was able to squeeze in their BDO product, which is perhaps a similar process and could take advantage of that situation. And that is very good, but it is uncommon. Normally, if you're running your plants, I can always run our fermentation facilities 98%, 99% full. You do not want to have spare capacity. You can always find some product to fill it in and go make money. So the trick is the way to get capacity is never by asking, do you have any free capacity? Is I have a product here which is more valuable than some of what you run in your tanks. Do you want to take a look at it? And absolutely, if there is something out there, and many of the companies discovering amazing new ingredients, we heard some of that today, or developing a brand or new applications of biotechnology that we and others have not conceived, by all means, we'll be happy to display some of the products that are being more commoditized in our tanks and increase the overall profitability of our asset. by putting in something more exciting. Usually, it works really well because of exactly what John said. Process science is really trapped in companies. So we've been doing this for 30 years. We have 300,000 liter fermenters. We have been doing precision fermentation for the longest time. And that knowledge is, you know, in companies like ours and a few others and ADMs and other players out there. So we can expedite those things for those companies, but moreover, we can make it economical. So in our industry, in industrial bio, it's different than therapeutics. Cost matters a lot. Manufacturing is deeply integrated with our research process, because if the cost ain't low enough, then there is no impact. You know, you can't sell it. The market doesn't take off. It's not enabling. So this is the equation to approach companies that have capacity. You know, I have a more valuable product here. Would you be interested? And usually the economics, they work in favor for both of us. You know, there's margin to be passed on both sides. I'll just finish by saying the one place right now, if I'm starting a company in industrial bio, not a therapeutics company, if I want to make some food protein that's going to need a lot of volume, and the price point is not therapeutics, we have an industry in the Midwest. We have the bioprocessing industry, the original, you know, 30, 40-year-old bioeconomy segment of the United States that's making ethanol and high fructose corn syrup. Those are two products that people no longer want. They don't want the sugar. They're not driving. When they drive, they want to drive electric. So you have assets which are fully depreciated with a very knowledgeable workforce that knows how to run fermentation, co-located to cheap sugar, right? And it's not going to work for every single product, but there's a lot of capability in those plants. And the value proposition there, it might be much easier to convince them I've got something better than ethanol or high fructose syrup to run in your tanks. And there's a lot of places where they will be happy to receive you.
That's an excellent point. And you may have seen recently that ADM just announced a $300 million commitment to a protein innovation lab in Decatur, specifically to help with scale into fermentation. The AB InBev announced an initiative, and I forget how much they're putting behind it, but it's a big initiative to make their fermentation capacity available. And then just last week, I was reading that Green Plains bioethanol, they are in the process of building 3 new plants specifically to convert their corn dry ethanol mills into biorefineries that can have the flexibility that you're talking about. And they actually acquired a company last year, Fluidquip, specifically to get some technology to let them do that. So, you know, your point is right on, that there is a great deal of capacity there. And if you're going to ferment it, you know, if you're going to use fermentation, it makes sense to have it close to the grain sources.
And Ron, I think we have Greg and Chris back on audio at least. We just had some bandwidth issues. So sorry about that.
Wonderful.
They should be able to hear you.
Yeah, how are we on time?
Go a little bit over if needed. I know we're coming up against an end. So take another 5 or 10 minutes.
Okay, well, we cut off Greg first, no, Greg second and Chris first. So Chris, why don't we go back and give you the mic, and then Greg will come back to you.
Well, I don't know whether you want me to try to go back through what I talked about or to comment on what's been said. So in the interest of time, pick one of the two.
Comment.
Go ahead.
Yeah, comment, please, Chris.
Oh, my comment. So I've seen, I hear people talk about repurposing ethanol plants. And one of my concerns there is ethanol plants, maybe it's us aseptic fermentation guys being snooty about it. But we like to refer to ethanol fermenters as big dumb tanks. They're cheap, they're not aseptic, they don't operate under pressure. And you have a very specialized process in there that's usually low pH. It's got ethanol, which tends to discourage other things from growing in there. Are there processes, food, for example, food process that will operate in a tank like that? Well, I know the citric acid fermentation that runs at a low pH, and that goes into a food product. But again, that doesn't have to go through the same sort of aseptic environment that other processes have to go through, just because, again, it discourages anything else from growing in there. So I have recently come across a company, Blue Stem Biosciences, that is actually addressing this issue, and they're trying to look at these bioethanol plants and what can we put in there that will work. In other words, let's take a look at anaerobic processes, and that can run in these big dump tanks under less than sterile conditions and produce something other than ethanol. And so I think this is a very innovative way of trying to approach the problem, and as an opportunity to repurpose an extensive amount of infrastructure. And hey, ethanol industry is so up and down price-wise that if you can come up with something that's a little bit more stable, I can, to the point earlier made, displace other things. Absolutely. I mean, displacement is always going to be, I think, a strategy, because that economics is going to dictate that. But the problem is, if you have new products, let's take Perfect Day Foods, or let's take every company, they want to revolutionize, the dairy and the egg segments. They're not going to do that by displacing existing capacity. They have to build some huge capacity. I've had the privilege of seeing some of these numbers, and we're not talking, a drop in the bucket. We're talking, many, 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 many buckets to make that happen. So I'll shut up with that.
Thanks, Chris. Greg?
Yeah, I agree with all the comments Chris and Luis and the team said. I just want to add, I know we're running out of time, so add a couple more comments. I think when you look at capacity and you look at what's out there, I think pre and post COVID, we were approached by many, many different companies to run someone else's product. And the first question we ask is, if you're going to utilize our real estate or the reactors, is the product better or than what we currently have in that reactor, right? So I think that's the piece that we always ask is, are we getting better and more efficient, or is it a better product coming out of that fermentation to displace that? I do agree that there's not a one size fits all reactor, by the way. So there's going to be CapEx put in whether you use ethanol reactors or all the reactors, but we do got to get better as far as the process R&D piece of it to get smaller footprint. And that's a piece where we're adding, hundreds of scientists in there in our process R&D to get the strain development right, the fermentation right, so that we're more efficient and we use less reactor real estate than we currently do. And that's the piece where I totally agree. We're not putting as much science and development in there as we should be. And that's a piece where we actually shifted, our resources. We have 1,700 people in R&D. We started shifting resources into the process R&D to actually get more efficient in more out of the reactor than we ever have before. Because putting steel in the ground is probably an old type of strategy, and we're trying to get more efficient in that matter. And I think that just goes with all technologies, getting the smaller footprint. So but I agree with the team, and I'll stop there. But Luis made some great points about what we're doing, and I think the process R&D needs more attention.
Well, let me thank the panel. Or do we have time for questions?
Well, I think we're running out, but we could.
Yeah, why don't we, we started late, and I think we should just keep.
Yeah, sure. We can start a little bit later, but yeah, let's go for questions.
What's your name? The new one that came out, Jeff? Is it Jeff?
Luis.
Luis Cascao. I'm here in Palo Alto with IFF. Yeah.
So I have I know very little about this world, and I've just learned a lot from the panel, but how much of the development side of things is really kind of like a product market fit issue, where so much energy has been spent on discovery mode, and now the market is ready to apply that and try to find use cases for it. And so now you need capacity in order to actually apply, and the market will probably catch up on itself maybe naturally, because there's also an end user that wants said product and so on. Does that resonate with anyone? But
I think it varies a lot by the field. I mean, the ratio of discovery spend and time for a cancer drug to development is very different than it is for a new food ingredient for grass affirmation. It's hard to have a specific answer, but I'll tell you my general impression. If my grandson just said, "Grandpa, what's the general assumption?" I'd say assume it's if you spend $1 on discovery, you're going to spend $10 on development. Maybe more.
Yeah, sure. Go ahead. Maybe just grab a mic right behind you.
So basically, the question is for the investor side of the conversation. And I think you also raised it that as a startup, you have two options. You have one option that you will finish the discovery, you start development, raise funds, you develop big facilities, you invest, and then you will go to the market. Probably fastest way. It's going to be the fastest way to go to the market, but it's a different model. There is another model that you also raised that the facilities in some case is already there, but they all belong to the giant companies. Now you need to set with them partnerships. Partnerships takes time, who belongs to whom, the IP, what I will get, what you will get, validation, who is going to sell it, what will be the value sharing, and etc. and etc. Now, the humblest way is to go to partnerships. The problem is that the growth of the organization will be slower, and also the evaluation of the company will be slower. Raise capital and building your own assets will be faster and also will bring a lot of promises. So how you define it as an investors, what you will prefer, or how do you define it down the road to the future? Because it seems that the more validation will occur, the more investment a startup will need to carry on to be in the market.
So I'll take a shot. I think you choose between those two models based on the economic conditions that you're facing as a startup. And you're facing some of the worst economic conditions we've seen for startups in the last decade. So the party's over. Money's expensive. VCs have their pockets zipped shut. They can sit out for 3 months. They can sit out for 6 months. I've heard of groups even thinking about sitting out for a year. That is not doing any investments for a year, only concentrating on their portfolio, making sure their portfolio companies are fine. When you find yourself in that environment, that's the time when you may start thinking about, maybe I'm going to go form a partnership to get this thing done. 3 years ago, you could have said, no, I'll just go to SoftBank or I'll go to somebody and I'll raise a big ton of money and go do stuff. And we saw lots of companies doing that. It's much, much tougher now. So I think it depends on the economic conditions you're facing and who your current investors are too. So.
Sure. So the panel is great, and the questions you laid out, Ron, were fantastic. So 2 quick questions. One is, I think that SynBio, it's just so broad. And so it'd be great to hear, there are areas of SynBio which actually don't require, $1 trillion of new infrastructure. So, thoughts on, if you were going to look for SynBio investments that actually make sense and don't require all this steel in the ground, what might those be, that would play out? So that's one question. And the other question is, I think in the food space is different in the ag space, but in the food space, predictive analytics haven't really been utilized nearly in the way that they could or should. And maybe in the, this precision fermentation space, what would, is there room for predictive analytics to come in and actually enable better decision making so that the right discoveries are moved forward, the right way, as opposed to humans just alone screwing it up over and over again?
Yeah, I can jump in there and add in. I think that's coincidental. We just had a presentation to our board today just on the digital piece of the analytics piece, and what's happening very similar to discovery and development is there's been a lot of data generated by discovery and a lot of information generated, and that's going to a lot of data lakes and data pools. But then to take that data and then take those analytics and then work that down into a process R&D so you get more efficient in that production hasn't had as much investment. And that's where we're looking towards more investing in that bottleneck. So bottleneck of taking that data, then translating that data into decisions to further refine that process R&D and fermentation capacity and strain development, et cetera, et cetera. Now back to your first question, what will we look at as an investor? One of the big areas that is high growth but utilizes less capacity is in fermentation is actives. For example, an active cosmetic area, which it's a high growth area, high profitability area, but it doesn't use that much real estate in the tank because a little goes a long way. The other one is in fragrance development. We've been looking at, for example, SynBio creating new fragrance molecules that obviously parts per million, parts per billion go a long way. So you're not chewing up as much real estate, and your growth area is double-digit growth per year. So those are two areas that at least my company we look at as in prioritization of the capacity.
I think in the fresh food area, gene editing, we've already seen one company since its founding in 3 years bring a product to market through gene editing. This is Pairwise and their conscious food brand, which has just launched a salad with a mustard green that's much better for you than kale, and it's got a deletion that removes kind of a bitter taste that it had, and so now it tastes absolutely fantastic. So that's in 3 years. Didn't need a lot of capacity, no steel in the ground. Yes, you've got to get growers on board. I think there's going to be a real revolution in the fresh food market thanks to gene editing and the favorable regulatory environment. So,
Well, with that, I think we should wrap it up. But Ron, thanks so
Well,
much.
then let me thank the panel very much and remind everyone that we're under charter host rules, which means I won't have to read a headline that says Meeusen predicts the end of discovery. Thank you.
Thanks. Thank you all. Thank you all very much.